Today we will discuss the physiological mechanism for the absorption of glucose. First, a carbohydrate such as starch is digested into glucose monomers by your digestive system. Then, for your body to use that glucose, it must be absorbed into the blood. At the small intestine is where glucose is absorbed into the blood. Let's diagram out a small section of the small intestine on the board, and we'll discuss this physiological mechanism for the absorption of glucose. This mechanism will recall on information you have learned previously in our membrane transport lecture and concentration gradient lecture. Here is a small section of the small intestine. These are the internal walls of the small intestine made up of enterocytes, which are epithelial cells. The space between these enterocytes is referred to as the lumen, and the lumen contains digested products such as glucose. Going back to these enterocytes, the side facing the lumen is referred to as the apical surface, and the opposite side is referred to as the basolateral surface. Surrounding these enterocytes are capillaries, which contain blood. Now, the goal for glucose absorption is to move glucose from the lumen of the small intestine through the plasma membrane of the enterocyte, through the interstitial fluid, and then into the blood. Now, from our membrane transport lecture, can glucose directly move through this plasma membrane by simple diffusion? No, because glucose is a large polar molecule. Remember, small uncharged polar molecules such as CO2, as well as nonpolar molecules, can move directly through plasma membranes by simple diffusion. So, in order to accomplish this goal, we need to rely on three carrier proteins. The first carrier protein is located on the basolateral surface, and this is a sodium-potassium pump. The second carrier protein is located on the apical surface, and this is a sodium-glucose cotransporter. And the third and final carrier protein is also located on the basolateral surface. This is a GLUT2 carrier. Now, all three of these carrier proteins will work together and allow for the movement of glucose from the lumen of the small intestine through the plasma membrane of the enterocyte and then into the blood. So, remember, what do sodium-potassium pumps do? Yeah, they take 3 sodium ions from the inside of the cell and move them to the outside of the cell against their concentration gradient, as well as they take 2 potassium ions from the outside of the cell and move them into the cell, also against their concentration gradient. Because these ions are moving against their concentration gradient, this process requires ATP. And this is an example of primary active transport. Now, because we're moving sodium to the outside of the cell, it's going to establish a higher concentration of sodium outside the cell. And thus, there will be a lower concentration of sodium ion inside the cell. Now, let's move on to our second carrier protein. This is a sodium-glucose cotransporter. So it's going to move sodium and glucose. And what it does is it takes 2 sodium ions from the outside of the cell. Remember, the concentration for sodium is really high outside the cell. And it's going to move this sodium from high to low because there's lower sodium ion concentration inside the cell. This process is favorable. In addition, it's going to grab onto glucose and also move it into the cell, but against glucose's concentration gradient. So thus, that must mean there's a high concentration of glucose inside the cell, and there's a lower concentration of glucose outside the cell. This is an example of secondary active transport, where the downhill movement of sodium from high to low concentration provides the energy to move glucose against its concentration gradient to the inside of the cell. As a quick analogy, your friend could be sodium. He has a lot of energy and he wants to go outside and play basketball with you. You, on the other hand, you're glucose. You've been studying for your physiology exam late last night and you have low energy and you do not want to go outside and play basketball with him. So your friend uses his energy to drag you outside with him against your will to go play basketball with him. This is what's happening with sodium and glucose here. Now that glucose is inside the cell, remember, glucose concentration is higher inside the cell and it's lower outside of the cell. Glucose, we want to move this into the blood. So we can move glucose from high to lower concentration through the GLUT2 carrier by facilitated diffusion. And that will allow glucose to move from inside the enterocyte through the interstitial fluid and then into the blood. Any questions on this mechanism so far? I know there's a lot happening all at once. Okay, so in summary, it requires 3 carrier proteins to move glucose from the lumen of the small intestine and then into the blood for absorption. We first started with a sodium-potassium pump, and through primary active transport, we established a sodium ion concentration gradient where there's higher levels of sodium outside the cell. Then, using the sodium glucose cotransporter by secondary active transport, we moved 2 sodium ions down its concentration gradient, and this provided the energy to move glucose also into the cell, but against its concentration gradient. Lastly, we took advantage of this GLUT2 carrier, and by facilitated diffusion, we moved glucose from high to low concentration through the GLUT2 carrier into the interstitial fluid, and then the glucose moved into the blood. From here, glucose will move through the hepatic portal vein and then into the liver. From our previous lectures, what are some functions or uses of glucose in the body? Yeah, glucose is an energy source. It's the primary energy source of the brain. It can be converted into many ATP through cellular respiration. Glucose can be stored as glycogen in the liver and skeletal muscles. Good. And yeah, glucose can be associated with lipids and proteins to make glycolipids and glycoproteins.